Shall we start, Dr. Bhatt?
Whenever
Let's go.
we should, okay. Good evening. I'm Tariq Bhatt, President of Chicago Medical Society. I would like to take this moment to thank you all for participating in this historic evening, important town hall meeting. We have an exceptional panel here to help answer questions. As many of you know, that Chicago Medical Society leadership and its COVID-19 task force, which is led by Dr. Vishnu Chundi, have been meeting on daily basis since the onset of pandemic, and now on weekly basis. And Dr. Johnson and Dr. Chundi have done a great job in leading us through this pandemic. And I would like to also congratulate everybody who has done such a wonderful job to bring this panel. But before I go there further, it's my pleasant duty to take a little time to introduce Dr. Steve Andra, who has been former advisor to President Obama and architect of Affordable Care Act and its many components. So without any further delay, Dr. Steve Andra, podium is for you. Thank you and welcome.
Thank you, Dr. Bhatt. It's a pleasure to be here and welcome everybody. This is part of our regular series by the Chicago Medical Society, intended to keep physicians as up to date and informed as possible during this extraordinary time. The next virtual meeting will be scheduled next week, and you'll get a memo on that after this. But tonight's meeting comes on the heels of a truly historic announcement. As I'm sure you've heard, the FDA panel recommendation is to approve an emergency use authorization for the first COVID-19 vaccine from Pfizer. Another panel is meeting next week. We expect the FDA will take their recommendation in the next, my guess is, 12 to 18 hours. And we'll look in the next week to next day or week to start distributing vaccine, but I'm sure Dr. Robinson will have more on that. It's really serendipitous that science and technology had us poised to take advantage of a new approach to vaccine development using messenger RNA at a time that the world really needed to have a much more rapid and efficient way to do that. But because it's new, there's a lot of questions. And so tonight, the goal is to inform the physician community a little bit more about how the vaccine was developed, its safety, efficacy, durability, and other questions, and how it's going to be delivered. I think that the people attending tonight have a real chance to take this message and propagate it out to your colleagues. We have hundreds of people on the call tonight, but there are thousands of physicians in the Metro Chicago area and throughout the country that you have access to, that you can take the information you get tonight and provide them with that to help physicians better understand how to care for their patients, how to inform their patients, and how to advise themselves, their family, and their patients on how to manage not only the vaccine, but this virus going forward during the peri-vaccine period. So we're going to take a look at safety, efficacy, durability of the immune response, and then from a public health point of view, how this will be distributed, what else does the public need to know, and what else do they need to do before, during, and after vaccination to help manage this pandemic. After the formal presentations, we'll have a question and answer session. Many of you have sent in questions. We've got those. We will run through those, have a chance for people to ask live. But now it's my pleasure to introduce the panel. As Dr. Bhatt said, we have a terrific panel. Dr. Ed Lin will be discussing the messenger RNA vaccine. Dr. Lin is a trustee of Chicago Medical Society. He's the former professor and interim chair of the Department of OBGYN at Rush University, Rush Medical College, my alma mater. And previously, he was chair at OBGYN at Cook County Health and Advocate Lutheran General. He's contributed to the scientific literature and the community in many ways. Dr. Dan Johnson will be giving us a summary of the FDA approval process for this current set of vaccines. Dr. Johnson is a nationally recognized pediatric infectious disease specialist. He's a professor of pediatrics at University of Chicago Medicine. He's a member of the newly formed COVID vaccine scientific work group of the Chicago Department of Public Health and is an advisor on COVID to Chicago Medical Society and advising school districts across the state. We're fortunate to have Dr. Candace Robinson, who will be wrapping up the formal presentation, discussing the vaccine distribution process. Dr. Robinson is in the Department of Public Health. She's a medical officer with the CDC. Graduate of Vanderbilt University School of Medicine. She has a distinguished career in infectious diseases and immunization management, as well as epidemic responses such as Ebola. Dr. Robinson is a leader in the National Center for Immunization and Respiratory Diseases in its immunization division. And lastly, I'd like to introduce Dr. Vishnu Chundi. He's a member of this panel who will participate in the Q&A session after the formal presentations. And many of you know Dr. Chundi is a specialist in infectious diseases and trained also at Rush. He's a senior partner at Metro Infectious Diseases, the largest ID group in the US. And in addition to his domestic work, he's deeply involved in healthcare in the country of his birth, India, where he's involved in HIV care through the Project SHARE. Most relevant to this evening's discussion, Dr. Chundi is the chair of the COVID-19 task force of the Chicago Medical Society, as Dr. Bhatt referred to. And with that introduction on what we want to accomplish tonight, we're going to have Dr. Johnson kick off the formal presentations. Then I'll be back as moderator to go through some questions and answers from this distinguished panel. And hopefully at the end of that, you'll all have a little bit better understanding that will help you manage your patients, yourselves, your families through this difficult time. Dr. Johnson.
Thank you very much, Dr. Andra, for the introduction and your opening remarks. And thank you, Dr. Bhatt, for your leadership of the society, and Dr. Chundi for leading the COVID work group. So thank you all for joining us tonight. May I have the next slide? As Dr. Andra mentioned, today was indeed a historic day. In fact, for those of you who celebrate Hanukkah, the holiday that starts tonight, it was quite a nice Hanukkah present to receive word from the FDA. Hopefully this will then translate into a successful vote by the ACIP, and we will begin to receive vaccine in many of our places. Now, of course, this was anything but a traditional process for the development of a vaccine in terms of timeline, but actually it was quite similar to the development of other vaccines, and I'll spend a few minutes going over that. What you see in front of you is the traditional vaccine development pathway that the FDA uses, and in fact, pharma uses as it develops a vaccine. And you can see that it's really quite a linear process, one that one step leads to the next, and to the next. And we usually run through animal studies leading into phase 1, then phase 2, phase 3 studies. It's quite linear in part because that's what the typical regulatory process expects. It's also linear in part because of the cost of creating a vaccine and the willingness for the type of dollars to be invested. Typically, the process takes somewhere around 10 years or longer. And you'll see with the next slide what really changed here. And you can see then that this is not as linear a process. And that's in part because of the assurances that have been made at the outset of this process, which allowed the more rapid collapsing of the process into what has amounted to roughly 11 months. And over this 11-month time period, there has been the assurance of dollars, which then allowed for processes to overlap. The animal data was being collected almost simultaneously with the human data, and the phase 1, 2, and 3 studies were really overlapping with each other, allowing a much more rapid planning process because the dollars were assured, both in terms of the dollars that would come from the federal government to underwrite some of the research work, but also the guarantee of purchasing of vaccine, not only by the US government, but worldwide, which gave assurance to industry that rather than having a long decision process occurring at every single step of this process, but rather the ability to be able to bring these steps into a much more collapsed manner. But what's really important is that phase 3 for this vaccine was as large, if not larger, than some of the other phase 3 studies that are used to give us the assurance that we need for vaccine development. Next slide. And so if we look at the emergency use authorization that the FDA operates under, I think it's critical first for all of us to know that this process came out of the 9/11 catastrophe and the desire to speed the rate at which potential new medical products can get to the public. It still requires a high level of standard, but the standard is a little bit different compared to the usual licensing process. Where here, the standard requires that we know that the effectiveness and the potential benefit outweighs any known or potential risk. And that there's no alternative product that the FDA can use that would be poised to be ready to be used. And certainly that applies to this vaccine. The other thing that was made clear in the process by the FDA was that any COVID vaccine would have to demonstrate a clear and compelling quality, safety, and efficacy in a very large, well-designed phase 3 trial, which is exactly what's happened here. The level of participants are on the 30 to 40,000 level, which is really very large and allows us to be able to make decisions. And then it would have to be reviewed by an independent advisory committee, which is what happened today. The other thing was that there would have to be enhanced post-deployment. And I know Dr. Robinson's going to talk a bit about that, so that way we can all see just how impressive the post-deployment surveillance process is going to be. And then finally, there was a requirement that there be at least 2 months of follow-up for efficacy and safety. And the reason for that is really quite simple. If you look at the history of vaccine development, after introduction into humans, literally within 6 weeks, we have seen all of the side effects, and that 6-week time period gives us assurance around quality. What it doesn't give us reassurance by, and if you will just advance for one moment, you'll see the last line of this slide. And what it doesn't give us is duration of efficacy. But already we have a good sense of duration of efficacy from some of the phase 1/2 data that's been generated. And we know that this vaccine is going to have efficacy also based on the immune response of the host to SARS-CoV-2 infection of at least 5 months and probably much longer. And with that, I'll turn things over to Dr. Lin to talk a bit about the technology of mRNA. Ed? You're muted, Ed.
My mute was stuck. I want to thank Dr. Johnson for his presentation at this point, and especially the Chicago Medical Society for putting together this very important conference. Hopefully, we'll be able to help you understand better some of the aspects of this vaccine that's coming out, and help reassure everyone that this is a very exciting kind of landmark development, which will be beneficial. May I have the next slide, please? So we're talking today about the mRNA vaccine. There are actually 2 in the pipeline for approval, as you're aware. The one that was given a positive vote today was the Pfizer product, which was developed in joint with BioNTech, a German company, and the Moderna vaccine, which we've heard about too, was a co-development with the National Institute of Allergy and Infectious Disease. So I just want to talk to you a little bit about what the difference is maybe between an mRNA vaccine and the more traditional vaccines that you're used to. Just think of vaccines as either being protein-based or gene-based. Now, protein-based vaccine would deliver the immune stimulating antigen to the host, such as a whole inactivated or killed virus. So we're very familiar with polio, the influenza vaccine, or we can have subunits of the vaccines and virus-like particles, such as hepatitis B and the HPV. Now, a gene-based vaccine is different in that it's simply implanting the genetic instructions to the host cell to produce the antigen, which is actually more like the natural infection would occur. In the case of the coronavirus, the antigen of interest happens to be those spike proteins, which we've all seen the pictures of the viral sphere with these projectiles, which are necessary for the virus to attach and to start passing the genetic material into the host cell. So in the way these are designed, if you look at this schematic, you can see that you have a virus delivery vehicle. And in the case of the product from Pfizer, it's a nanoparticle encapsulating the mRNA. It enters the cell, and in the cytoplasm, it's releasing the mRNA, which then is going through the cellular machinery for the production of the desired protein, which in this case is the spike protein. So it's, in essence, we're giving the genetic material. The host cell then is going to be producing the antigen that's needed for the immune response. Now, this approach is not necessarily an unfamiliar approach to those who are thinking about vaccinology, because a live attenuated vaccine, such as the measles, mumps, rubella vaccine, you will incorporate the genetic material into the cell, and it programs the cell to then produce copies of that virus, and then antibodies and a T cell response would subsequently occur. In early 2002, the SARS and the MERS infections, which were coronavirus infections, stimulated a lot of research over the subsequent 10 years. And the focus then was pinpointed onto the spike proteins. And a big advantage, because unlike the conventional vaccines, the mRNA vaccine is not grown in eggs, not grown in cell culture. These are simply chemically catalyzed in the laboratory for the easy development, which in this case led to a quick development of this vaccine. And it's also more easily scalable to produce massive amounts of the vaccine for distribution. May I have the next slide, please? Now, in the Moderna vaccine, it will work the same way. What I want to point out in this slide is actually pointing out maybe some of the advantages of an mRNA vaccine. The genetic approach has the potential to be more advantageous immunologically. and let me point this out. As you know, antibodies are produced when the viral antigen, this protein, gets into the circulation or into the lymph nodes. But also very importantly, is it allows the production of the CD8-4 T helper cells and the recruitment of CD8 cytotoxic T cells through a different pathway, as you can see to the right of the graphic here, the Mhc pathway, which is the major histocompatibility pathway. So one of the advantages of the RNA type vaccines is that we will get immune response, both humoral as well as cell mediated. And actually it's that cell mediated immunity that may be responsible for the longer duration of effectiveness. So I hope I've clarified very quickly for you a very complex bit of basic science. And now we can move on to discuss a little bit more of the clinical importance as we're faced with the imminent release of the vaccine. I'd like to turn things over to Dr. Cynthia Robinson. Candace Robinson, I apologize.
No problem. Thank you, Dr. Lin. Good evening, everyone. We get started. We're going to talk a little bit about kind of timeline, a little bit about the Pfizer vaccine, Mrna vaccine, and then a bit of information about the rollout plans from CDPH for the vaccine here in Chicago. Next slide. So as quickly as I make slides, they're almost outdated. So as you can see, this slide, which was made last night, talked about the fact that the FDA would be meeting today to discuss the Pfizer vaccine. And so as we heard at the top of the presentation from Dr. Andra, the advisory committee voted to approve or voted for the EUA, and the FDA will do their paperwork from there. And we are anticipating still, however, that the FDA VRBPAC committee will meet next week on the 17th to discuss the Mrna vaccine. Next slide. And so what happens next? The ACIP has actually announced that they'll be meeting on Friday and Sunday to continue their discussions on the Pfizer vaccine, and they have scheduled a vote from the ACIP for Sunday. So if the ACIP does in fact approve the vaccine, then we expect first doses of the vaccine to be arriving in the city sometime next week. And then we would follow a similar pattern for Mrna, provided the FDA authorizes the EUA. The ACIP would then meet kind of in the weekend after that, and we could see the first doses of Mrna vaccine arriving here in Chicago the week of the 21st. Next slide. Let's talk a little bit about each vaccine. So the Pfizer vaccine, I'm sure we've all heard a lot about it requiring ultra cold temperatures for storage. That's somewhere between -80 and -60 Celsius. And then if the vaccine is thawed and stored in the refrigerator, it then must be used within 5 days. And once it's reconstituted with a diluent, which it requires, then it's a matter of hours in which the vaccine must be used. And so because of the storage requirements, Pfizer is distributing the vaccine instead of through a distributor, like with many other of our vaccines, they're actually be shipping it directly from the manufacturer to the facilities. The Pfizer vaccine is a 2-dose series, and both doses dose 1 is recommended, and then dose 2, 21 days later. And the minimum order amount for this vaccine currently is 975 doses. The federal government has kind of put out some estimates, and they expect somewhere between 20 and 30 million doses of this vaccine will be available in the US by the end of the year. Next slide. The Mrna vaccine has storage temperatures that are -25 to -15 Celsius, which is closer to, of course, our standard storage temperatures for MMR and varicella vaccine. If this vaccine is thawed and stored in the refrigerator, then based on the information that we have to date, then you would have to use it within 30 days. This vaccine will be shipped through a distributor similar to vaccines under the Vaccine for Children's program. While this vaccine does not require reconstitution, it is a 2-dose series as well, with doses separated by at least 28 days. And there's been some estimates from the government that we could expect potentially approximately 15 million doses of this vaccine to be available in the US, of course, pending FDA and ACIP. Next slide. So here was some of the data from the FDA briefing documents that were put up by the Pfizer on the website prior to the FDA's discussions today. So in these briefing documents, it talked a bit about the efficacy, and the efficacy in preventing confirmed COVID-19 was approximately 95%. And that was based on 8 cases in the vaccine group and 162 cases in the placebo group over the same time period. And sub-analysis shows similar results across age groups, gender and racial ethnic groups, and the participants that they had in with medical comorbidities associated with high risk of severe COVID-19 disease. Next slide. You know, there's still some things we don't know about efficacy, and we'll have more data coming. So for instance, the duration of protection, efficacy in young children, efficacy against mortality, efficacy against asymptomatic infection, and efficacy against transmission. And we'll get more data on this over time as the vaccine is used as we go along. Next slide. A little bit about safety. So the available safety data from the 43,000, over 43,000 participants that Pfizer had enrolled in their trial through November 14th, with a median follow-up of 2 months, did not raise any serious safety concerns. And so the briefing documents noted that the known and potential benefits of the Pfizer vaccine, this is the question they considered, and this is the question that actually the committee voted on, was whether or not the known and potential benefits of the vaccine outweighed its risks for use in individuals 16 years of age and older. Next slide. So the side effects from the Pfizer vaccine, for some people, the side effects are more noticeable than the side effects from other adult vaccines, like the pneumonia vaccine or flu vaccines. Side effects usually lasted a day or two, and they were more common, according to the briefing documents, after the second dose of vaccine rather than the first dose of the vaccine. Next slide. So this shows local reactions data for after the second dose of the vaccine. Approximately in the briefing documents, they highlighted that approximately 78% of those 18 to 55 had some pain at the injection site. And then over the age of 55, interestingly, that symptom was slightly less at about 66%, and about 4 to 7% reported redness and swelling. Next slide. And then for the systemic side effects after the second dose of the Pfizer vaccine, 59% reported fatigue. Interestingly, there was about 23% for the placebo, 52% reported headache, 37% reported muscle pain, and 35% reported chills. And interestingly, these side effects were less common in people over the age of 55 as opposed to those 18 through 55. Next slide. Another thing that often comes up in discussing vaccines were deaths during the trial. And so there were a total of 6 deaths of the 43,000 participants during the trial. Of those, 2 were in recipients who received the vaccine, and 4 were in persons who received the placebo. And then for both vaccine recipients, both were over the age of 55. One experienced a cardiac arrest and the other one had arterial sclerosis. And then the placebo recipients had reported different causes of death, such as myocardial infarction, stroke for one, and then unknown causes. And 3 of those 4 deaths were in the older age group. And important to note that all deaths represent events that occurred in the general population of that age, where they occurred at a similar rate. So there was no signal that this was increased in either the vaccine or the placebo group. Next slide. And so some things we don't quite know yet: safety in certain subpopulations like young children, adverse reactions that are very uncommon or that require longer follow-up to be detected, and further signal detection efforts for these adverse events will take place with more widespread use of the vaccine. And I'll talk here momentarily about kind of the robust systems that are in place for safety monitoring. Next slide. So there are multiple systems that will be going on for ongoing safety monitoring. vSafe is a new active surveillance system put in place by CDC. I'll talk about that a little more in depth here shortly. VAERS, the Vaccine Adverse Events Reporting System, is the one we're all probably very familiar with. It's the passive surveillance reporting system. And then, of course, they will continue to use CISA, the Clinical Immunization Safety Assessment Project, for individual case consults. And then there'll be kind of large linked database monitoring as well, so the signals can be detected through systems such as the Vaccine Safety Data Link. Next slide. So I'm going to focus specifically here on a couple of these, vSafe and VAERS. So VAERS, of course, is one we are very familiar with. We use it for all of our vaccine adverse event reporting currently, and it's the nation's early warning system for vaccine safety. So this is co-monitored by CDC and FDA. They typically receive, somewhere between 40 and 50,000 reports a year. And it's important to note, of course, that VAERS reporting does not necessarily mean that that event was related to the vaccine. It's kind of where they collect the data and they analyze it and determine, if there's a signal there that needs to be investigated further. And then more information and how persons can report to VAERS is at the VAERS website. Anybody can report to VAERS, the provider, the patient, et cetera. And it's important to note that part of the CDC's COVID-19 vaccine provider agreement stipulates that providers have to report any adverse events, whether or not they think it's related to the vaccine, to VAERS. Next slide. The new system is called vSafe. And so once the vaccines are made available, in addition to these systems that are already existing, like CISA, the Vaccine Safety Data Link, and VAERS, there'll be a new system, vSafe, to monitor vaccine safety. And so vSafe is a smartphone-based tool. It'll use text messaging and web surveys to provide kind of personalized health check-ins after someone receives the vaccine. And so through vSafe, vaccines recipients can quickly tell CDC if they've had any side effects from the vaccine. And depending on their responses, CDC may follow up with them by phone to get more information. And vSafe will also remind them to go get their second dose of the COVID-19 vaccine for those that require 2 doses. Next slide. So the participants who enroll in this program will receive daily texts for the first week post-vaccination, and then weekly thereafter until 6 weeks post-vaccination. And then there'll be additional health checks at 3, 6, and 12 months post-vaccination. And then this timeline resets after the second dose. So they'll have kind of separate timelines going on for the first dose versus the second dose. Next slide. And so part of this is that CDC, of course, is asking that when persons receive vaccines, that they kind of get this flyer and get this QR code that they can scan. And once they scan that QR code, or they can sign up on their mobile phone via the browser, they will be enrolled in the program and they will start to receive those messages. Next slide. And so I think some of the side effect messages for this, for most people, side effects are mild and go away on their own in a couple of days. But I think we also need to let people know about, the local reactions and things that can occur, particularly after the second dose, because, having people be aware of that ahead of time, then they won't be surprised. I think it's similar to kind of the education we've been doing around the Shingrix vaccine, where you can have some enhanced reactions following that vaccination, and making patients aware of that ahead of time will be helpful so that they know what to expect and that they won't be surprised when it happens. Because if they aren't informed, then if they have a local reaction or they have a systemic reaction following vaccination, then they might think, oh, something, I didn't know this was supposed to happen. And so them being informed is the best way. And then, of course, the side effects for messaging for patients. Side effects do not mean they have COVID-19 vaccine, even though some of the symptoms may be the same. You cannot get COVID-19 from this vaccine. As Dr. Lin talked a lot about how the vaccine works, you can't get the virus from the vaccine, which is another myth that we've been hearing recently. No serious safety concerns have been observed in the clinical trial. And I think another important point to highlight is that, COVID vaccination will be a safer way to help build protection. COVID itself, of course, can have serious life-threatening complications, and there's really no way to know how COVID will affect someone individually. And of course, if someone gets sick, they could spread it to their family, friends, and those around us. I'll highlight our website here where we have other FAQs and other information that providers can access for answering patient questions. Next slide. So one thing to note about COVID-19 and vaccine distribution is that in the initial phases of vaccine distribution, the amount of vaccine will be very limited. And then over time, we'll continue to have increased doses of vaccine available as more doses are manufactured and as more vaccines potentially come onto the market. But in these initial phases, the vaccine supply will really be constrained and be limited. Next slide. And so that requires vaccine prioritization plans for vaccination in these early stages when the vaccine is limited. Now, the Chicago Department of Public Health are basing their plans on national guidance. A couple of months ago, the National Academies of Sciences released their framework for equitable allocation of COVID-19 vaccine. And just last week, the ACIP actually met to vote on their first group that they recommended for vaccination. Both groups included healthcare personnel as the first population recommended to receive vaccine, but there won't be enough vaccine to vaccinate all healthcare personnel immediately. So there will need to be some sub-prioritization even amongst healthcare personnel. Next slide. We get this question a lot, who's considered healthcare personnel? It's really a broad definition. The CDC defines it as anyone paid or unpaid serving in healthcare settings who have potential for direct or indirect exposure to patients or infectious materials. So they really encourage facilities to think broadly about who this includes. And this is everybody from our physicians, nurses, pharmacists, students, trainees, environmental services, security, et cetera, administrative, et cetera, could all fall into that category. Next slide. So this is just the chart from the NASM, the National Academies Framework. As you can see there, they had phase 1, high-risk health workers and first responders. And then additional phases for as vaccine supply increases. Next slide. And the ACIP voted only on this phase 1A group, but they voted to include long-term care facility residents in this first group who are recommended to receive vaccination. Next slide. So, the Chicago Department of Public Health has been planning for vaccine rollout, and in these initial phases, focusing on both the healthcare providers and the long-term care facility residents. So we'll be making direct allocations to hospitals and health systems for the vaccination of their employees, but we're also making plans for vaccination of staff and of clinics and facilities that are not associated with hospitals or health systems. And so to assist with that planning, CDPH has distributed a survey where one member of representative from a clinic or facility can complete the survey for staff vaccination options for persons in their clinic or facility. And then CDPH will reach out with vaccination options. So we're planning for both those in hospitals and the outpatient providers or other providers who may not be associated with a hospital or health system. And then as the supply increases, we'll expand our vaccination efforts based on national level prioritization guidance. Next slide. So we have ongoing communications for providers through situational awareness webinar calls that are happening bi-weekly, and probably for the next few weeks, they'll be weekly, just giving this pace at which things are moving. We've got task-specific communications that we've put out. For example, for providers who want to enroll to become COVID-19 vaccine providers, there's communications out on that. And really where you can find all information for healthcare providers related to COVID-19 vaccine, vaccination options, what calls we're having, instructions for becoming a provider is on our new Han COVID-19 vaccine site, which you can link to from the Han COVID-19 site. We also have our provider communications team. We've established a hotline, and we have a mailbox specifically dedicated to COVID-19 vaccine questions. Next slide. So for the successful rollout of the vaccine, CDPH has been thinking about this kind of from all aspects. The team, the logistics of the staffing and vaccine administration, either directly or through partners or through other clinics and outpatient clinics. The temperature, that's kind of the logistics of the vaccine itself. As we noted, vaccines like the Pfizer vaccine require ultra cold storage, and so there are logistics associated with that. Transportation, that's kind of the actual distribution of the vaccine. Training, given that these are new vaccines and they all have different storage and handling procedures, there's going to be a lot of training related to the use of these vaccines. Tech solutions and transparency, really working on the communications, both for our providers and for the public as we discuss these vaccines going forward. Next slide. So the CDPH is planning for kind of mass vaccination clinics as well. We used this model during our flu campaign this year and have validated kind of 4 different models that we will be using. We'll have our mass vaccination sites or points of dispensing, pods, and these will be kind of static sites that are operated on a regular schedule. And then we have mobile sites that we use for targeted penetration in a designated area or community. These sites typically hold a smaller volume of patients, but will be used and will be a medium through which patients can access this vaccine as we have more supply and as the groups that are recommended to receive it expand. We have a drive-through option that we are prepared to deploy, of course, not in January in Chicago, but in the spring or summertime. And then we have strike teams, which are typically location or outbreak specific response for vaccination. Ultimately, you know, we think the city will vaccinate directly around 250,000 plus Chicagoans. But of course, there are over 2.5, 2.6 million people here in the city of Chicago. And so really we'll be needing to work with all of our partners to vaccinate. Next slide. And so we'll be working, of course, with pharmacies. There's actually a federal program that partnered long-term care facilities with pharmacies for vaccination of long-term care facility residents. And we'll be activating that program to vaccinate that group as part of the phase 1A recommendations. There are federal expansion of a pharmacy partnership kind of when we get to phase 2, and we have expanded vaccine supply. We'll also be working with federally qualified health centers and other outpatient providers. So outpatient providers who are interested in becoming COVID-19 vaccine providers would have to enroll as a vaccine provider. And as I mentioned, information about that is on the Han website. And additionally, of course, we'll continue to work with hospitals and health systems as we expand, as we get more vaccine supply. They will move from vaccinating their staff to vaccinating the groups that are recommended for vaccination as we proceed through the phases. Next slide. So where can you go for information? We have the COVID-19 vaccine website, which is chicago.gov/COVID-vax. And this is where you'll find all the information, frequently asked questions, talking points, information for talking to patients about the vaccine, information for talking to anyone about the vaccine planning efforts we have in Chicago, information that addresses kind of some of the common concerns we've been hearing, all can be found on this website. Next slide. And we also have other communications. We have what we call message maps, which kind of discuss different topics that come up regarding vaccine efficacy, vaccine safety, et cetera. And these maps really work by kind of having a high level message and then providing the supporting information that informs that high-level message. And these will be useful in discussions with patients, but I think it will also be useful in discussion amongst us healthcare personnel, because I think we could potentially set the tone for vaccine uptake going forward as vaccine is expanded to other members of the community. Next slide. And then the CDC, of course, has multiple resources, FAQs, et cetera, through their coronavirus vaccine website as well. And this will be another place of information to get additional information. I think that's the last slide. Yes.
Well, thank you, Candace, and thanks to the panel. Just something I wanted to start off the Q&A with is lots of information that came to you. This session is being recorded and will be available on the Chicago Medical Society website, so you can go back to pick up things that you haven't seen. Also, I've been watching, there's an active chat that's been going on with the attendees to this meeting, as well as the panel. Lots of good information there. I don't know if the Chicago Medical Society has the capacity, but some of this Q&A may be distilled down and put on the website too. Let me give an assignment out where I have no authority to, or reason to do. In the last few minutes, we got 10 minutes for Q&A. I did want to say that all the sites that Dr. Robinson talked about and others, Chicago Medical Society as a site, are great sources of information. We all joked in medical school about the Journal of Irreproducible Results. There's so much misinformation out there that going to these websites, the Chicago Medical Society, Chicago Department of Public Health, and other vetted websites is your best way to get information to help your patients and yourselves. So with that, I've got a few questions for the panel that we have time for. And the first is this question about mutation. How long does the messenger RNA hang around? Can it reproduce? Does it get incorporated into the genome? There is a lot of myth out there. And can the panel talk about specifically those issues and concerns? Maybe Dr. Lin, since you started with the science, you could start us off. And Dr. Chundi, you can get in here, and I've seen you on the chat.
So again, I want to go back to the concept that this is a fragment of mRNA that was created in the laboratory to code for the production of the portion of the spike protein, which is the desired antigen for the host response. Whether it's the type of nanoparticle, whether it's in a lipid, lipoprotein envelope, if you will, or possibly another type of design, because there are other vaccines that are being designed slightly different, with different ways of introducing the material into the cell. And once it gets in the cell, it rapidly will do what it's programmed to do and produce the protein. Unlike DNA vaccines, which have to be incorporated into the nucleus and within the whole cellular apparatus, and that's somewhat inefficient because it only occurs during cellular division. This one works in the cytoplasm. And the other thing is that the portion of the protein, the spike protein, which is being coded for, happens to be identified as being stable, and there's not a lot of mutagenicity. I will allow my other learned colleagues to chime in on their opinion, because I think there's low probability that this is going to be an issue.
Okay, and Dr. Chundi, let me give you some yes, no's. Can this messenger RNA replicate in the cell?
It does replicate in the cell, but it degrades over time. That's why you need 2 doses of vaccine. And durability, we don't know yet. And when you will need a booster dose, we don't know yet, but we will over time.
Does it incorporate into the genome? Any possibility?
Absolutely not. It's the cytosolic. It goes to remember the old Golgi apparatus and makes protein that your body then makes antibodies to fundamentally, and the T cell immunity seems to be kind of key. So you have a long memory T cell for this vaccine. Again, the duration is going to be known as we give more of this vaccine.
Other comments on those key questions?
I'd just like to say these two vaccines are two separate proteins. The Mrna vaccine encodes for a different protein as compared to the Pfizer vaccine. And as far as I know, they're not interchangeable. Dr. Johnson, can you comment on that?
I agree, Dr. Chundi. The recommendations are whatever you start with, you use as the second dose. We're not aware of any interchangeability of these. Maybe eventually data will be accumulated in that regard, but it would require another trial. So at this time, whichever one you're given is what you should get as the second dose.
And Dr. Robinson, can you opine on this whole question of, does this make you immune from getting the disease or just being symptomatic of the disease? And if you get the vaccine, have a successful antibody response, do you have a chance of picking up virus and passing it to someone? And related to that, how long are we going to be in masks?
So these questions are kind of interconnected because we don't quite yet know have data for kind of, can you still transmit the virus after you're vaccinated? And so we're going to be learning more about that over time. But I will note that, you will still need to wear your masks after you get the vaccine, kind of in this peri-vaccine period. We'll still need to wear masks. We'll still need to social distance, et cetera, while we learn some of these additional things and until we kind of get enough of the population vaccinated. So how long we stay in masks will kind of depend upon two factors. How many people we get vaccinated and what herd immunity level we can achieve. And then it'll also depend upon disease transmission in the community and how much disease is spreading. So there are a couple of factors that will determine how long we're in masks, but we will still be in masks even after this vaccine comes out. And so don't throw your masks away yet. You will still need them for the time being. So
I've got time for a couple more. Dr. Chundi, Dr. Johnson, can you talk a little bit about, I think I might have had COVID in the past, and should I get the vaccine if I've had it?
Dr. Johnson, do you want to take that one?
Sure, happy to do so. People who had known or active COVID were excluded from the trials. At this point, we don't know the advisability of immunizing individuals who had recent COVID or were symptomatic from COVID at the time of vaccination. But there were people who were vaccinated who had antibody evidence of prior COVID, and that data is still being analyzed to be able to answer the question. However, we do have the immunogenicity and reactogenicity data, and what it shows is that there were no serious side effects other than the local effects and some very short-lived systemic side effects, none of which were in any way life-threatening. What that suggests then is that it is safe to immunize people who have had COVID previously. The current recommendations are around whether or not you should prioritize immunizing those individuals. And right now, the recommendation is that you should likely hold off immunizing anybody for at least the first 90 days after having acquired COVID, simply because the antibody response is so robust in those individuals and the immune response is so robust that there's no reason to immunize that group of individuals. Now, beyond 90 days is an open question, although if we look at actually data that came out in the last couple of weeks, it would suggest that immunity lasts for at least 5 months in the overwhelming majority. And in fact, the risk of reinfection is probably on the order of 0.001%. So there's probably no reason to immunize even out at 5 months. But since there have been some cases of reinfection at around 90 days, you may make the offer to a patient to be immunized. What I would expect is that those who have had recent infection are more likely to have a higher rate of local reactions and maybe have some of the systemic reactions of fatigue and fever. So that's the reason why I would also advise delaying immunizing that group for at least 90 days. Dr. Chundi, anything to add?
Well, the question I have for Dr. Robinson is, would you recommend getting antibody testing? Because not everyone who gets infected develops antibodies.
You know, the ACIP will weigh in on that, of course, but in similar situations, they have typically not recommended antibody testing before vaccination. For instance, if somebody doesn't have, you know, evidence of immunity to healthcare providers for certain things like MMR, like measles or Hep B and things like that. But we'll have to kind of wait and see what they weigh in on this. It'll be interesting to see their thoughts on this on Monday. So more to come on that.
All right, and we're going to go a few more minutes since we got a little bit of a late start. Dr. Lin, something in your wheelhouse. What about women of childbearing age? You know, should they get a pregnancy test? Was there any data in the study on this? And what about a woman who knows she's pregnant?
Well, unfortunately, women who were pregnant or even lactating were not actively recruited as participants in the studies. But we do know that there were women who became pregnant who were enrolled, and we're waiting for some more information. Now, if you look at the natural course of this disease in pregnant women, the Society for Maternal Fetal Medicine has pointed out that they can have a stormy course. And therefore, they feel that the risk-benefit ratio, based on what information we currently know, although it's limited, favors women getting vaccinated, especially if they have comorbidities. And that would, again, it's individual decision between the patient and her provider, because we really don't have robust data to be able to draw these conclusions. In terms of lactation, I think we feel a little bit more comfortable there, but there has not been any real problem with this in patients who had naturally occurring, COVID infection. And again, it's a personal decision, but it is not being actively discouraged. We're just hoping that, and we kind of wish, but hoping that more patients would be recruited for subsequent studies so that we can have more authoritative opinion on this.
So thank you, Dr. Lin. Dr. Chundi, since many COVID infections are asymptomatic, would you recommend getting antibody testing before a vaccine or not?
No, I don't think that's a feasible thing for a large population such as this, and the side effects are minor, basically local side effects. So I think people should just line up and get your vaccine. The other thing is, as healthcare, we work in the hospitals, we see a lot of pregnant nurses and doctors, and they're putting themselves at risk because they're seeing a lot of COVID in the hospitals. And so the risk benefit, like Maternal Fetal Society said, I agree with what they're saying, that these people should be offered vaccine. And if I was in their position, I would advocate for them getting vaccine, unless they're going to get away from the COVID population within the hospitals. And we have very few healthcare workers out there.
Thank you very much. We've all heard a little bit about a couple of people in Britain having severe allergic reactions, and it's been suggested that people with a history of allergic reactions not get a vaccine. Can you clarify what is a severe allergic reaction, and in which patients with a history of allergy should not get vaccine versus should?
Dr. Chundi, would you like to handle that one?
Yeah, the person in Britain, the two people in Britain who had a severe reaction, one had severe urticarial reactions in the past, and the second person had multiple allergies. And both of them had EpiPens with them because they were aware of their allergies. So I would suggest that people who have severe urticarial reactions and severe urticaria in general, be very cautious with the vaccine and probably have an EpiPen with them when they get the vaccine, because we can see the amount of morbidity and mortality with this virus. So I would certainly be careful giving them the vaccine, but I wouldn't preclude them from getting the vaccine. Dr. Robinson, do you have any comments on that? Since
Yeah, I would note that I anticipate that ACIP may discuss this and they may have some specific recommendations. So stay tuned to ACIP on tomorrow and Monday, because I anticipate that this may be a topic of discussion and they may make some specific recommendations. But stay tuned, we'll come back with that when they do.
Well, as we wrap up, there's many, many more questions. And fortunately, this is part of a series the Chicago Medical Society will be doing. I tried to rapid fire the questions as best I could, and it puts the panel in an unfair place of trying to give black and white yes, no answers to things that is all gray. So thank you for your patience with that. I would ask that the physicians that are participating in this session become ambassadors of real information to their colleagues, pass along these websites, and recommend that their colleagues take a look at this recorded session. So I want to thank the panel, thank the Chicago Medical Society for being a great source of information and ongoing information in this evolving story. Thank you for having me. And before we sign off, I would ask, does anyone from the Medical Society or Dr. Bhatt want to wrap up? If not, anyone from the panel with some parting words of wisdom?
Natalie has unmuted me because I was on mute. I just wanted to thank the panel again and Dr. Stevandra on behalf of Chicago Medical Society. This society will continue to bring very valuable programs. I'll encourage everyone to become members, acquire information, be ambassador of that information, as Dr. Stevandra mentioned. So thank you so much, and we'll continue on.
Thank you all for being there every day for the patients.
Thank you.
All right.
Good night, everyone.
Good.